Welcome to Unit 5.2 on chromosomes and mitosis. I'm Wendy Nofke and I'll be doing your presentation today. When we look at the genetic material inside of a cell, we often picture chromosomes. Chromosomes are actually a very specific form of the genetic information. This is the coiled form that the DNA will take during cell division. Here everything is very tightly packaged up to form the chromosomes that we see during this phase. The only time we're going to see chromosomes is during cell division. The rest of the time you want to have the DNA loosely spread out so that the enzymes can get in and reach the DNA and you can make use of that genetic information. The loosely coiled DNA that you're going to see during the non-dividing phase is chromatin. When we do have chromosomes, they're going to be made up of two sister chromatids. These are the two identical copies of the replicated DNA. When you picture the X shape of the chromosome, each of the chromatids is one side of the X. The centromere is the region where those sister chromatids are going to be attached to each other. Genes are going to be what actually contains the instructions for making a protein. These are located on a chromosome. So when you look and you have the legs of the X on the chromosome, just a small portion of that leg is going to be one of the genes. When cells divide, you're going to have homologous chromosomes that are going to be paired chromosomes of the same size, shape, and with the same genes. By pairing up the homologous chromosomes, you can ensure that each of the new daughter cells will have an exact copy of each of the chromosomes. Mitosis actually has 4 phases to it: prophase, metaphase, anaphase, and telophase. During prophase, chromatin is going to form those chromosomes. The nuclear membrane is going to start to break down, and the centrioles are going to begin to separate. When the centrioles are separating, this is going to help to start form the mitotic spindle that's going to help to pull things apart in the cell. During metaphase, the sister chromatids are going to align along that mitotic spindle. They will line up along the center. During anaphase, the sister chromatids are going to separate and move towards the opposite poles of the cell. And finally, during telophase, the chromosomes will return back to chromatin. So we uncoil that DNA so that it can be used in the cell again. You'll form a new nuclear membrane and the mitotic spindle disappears. This slide here on the left, you can see we have interphase here where the DNA is in the form of chromatin, nice and loosely coiled. And we move into prophase. Here you have chromosomes forming. You're starting to see the nuclear membrane disintegrate. During metaphase, you have these chromosomes aligned along the mitotic spindle in the center. During anaphase, you can see that these chromosomes are pulling apart to the opposite poles of the cells, and during telophase is when you're starting to see the cell begin to pinch off to form 2 new cells. Cytokinesis is the division of the cytoplasm. This process is slightly different in animal and plant cells. In animal cells, the plasma membrane is going to pinch inward along the center of the cell, along its equator, so that you can pinch off into 2 cells. With plant cells, you have a cell wall that's going to prevent that from happening. So instead of pinching, you're going to have a cell plate form along the equator. You'll create a new plasma membrane and cell wall along each side so that you create 2 new cells without having any pinching off.